Thank you, Waleed. My name's Lucy, and as Waleed mentioned, I'm a PhD candidate over at the University of Washington, and my department is a bit of a mouthful. We usually shorten it to BIME. We study informatics and medical education. And thank you all for inviting me back to give this talk today and discuss some of my research pursuits in the past couple of years and also maybe in the future. And as a little bit of background about me, I study biomedical ontologies and knowledge representation, and also how to apply these things to try to derive disease and disease treatment targets for various conditions. So that's what you're going to be hearing a little bit about today, and I'll try to clarify. I'll try to give background, enough background for you to understand this work, but if there's any questions, please feel free to interrupt during the presentation. I'll try to answer those before. So the title of my talk is Learning from Biomedical Knowledge. I'm going to discuss the creation or the curation and creation of some ontological resources and also how I've applied them in recent work. So to start, I'll give a little bit of background about kind of the history and state of biomedical ontology and why they're important. I will then discuss some aspects of my research, some of the projects I've worked on, and also some future work that I'm looking forward to doing. So to start, James Forleo and probably a number of other people have said this: health is simple, but disease is complicated. And the reason here is also complicated. I guess when we're healthy, we don't really think about trying to figure out the mechanisms that make us healthy. But when something goes wrong with our system, we really want to know what causes that. And it just turns out that the biological mechanisms that make us function are very complicated. So trying to tease out what happens when a disease strikes is a challenging problem. And I think structured biomedical data is really key to understanding how things go wrong in disease. So on the right there, I have the word phenotype. So that's a trait that is observable. So diseases count as phenotypes, things like chest pain or degenerative brain disorders or cancer. These are things that are observable changes to our body. But phenotype also includes things that are not necessarily disease states, things like height or eye color. And these things, we generally think of them as being directly linked to our genotype, or the genes that we inherit when we're born. But truthfully, it's a little bit more than that. There's also a lot of environmental factors and then some aspects of randomness. But genotype and environment, those are the things that we want to focus on because those are the things that we can measure. With genotype, we can measure that by sequencing our genome and figuring out what's there. Environmental causes, although we can't measure that directly, we can measure the effects of the environment on the way our genes are expressed in the body. So these are things that we can actually run experiments to sort of measure. So then it really started with the Human Genome Project. That was, we thought that by sequencing the genome, we would be able to understand how our genes contribute to phenotype. And the Human Genome Project was started several decades ago and was completed actually only in 2003, which was about 15 years ago. So we've had 15 years to study that data. And some of the earliest ontological resources were also created kind of in step with the Human Genome Project. So for example, the gene ontology, which is one of the earliest ontologies in the biomedical domain, was created mostly to annotate the genome. But of course, it's not quite as straightforward. It's not like each gene is necessarily responsible for one function in the body. And in fact, one gene can be responsible for hundreds of functions or thousands of functions in the body. So since then, there have been many more structured terminologies that have been created to fill additional voids in biomedicine. So things of importance might be like the cell ontology, which discusses different cell types, because it turns out that the same DNA that you have in your liver cell is not going to produce the same proteins as it does in your neurons. And then something like UniProt, which identifies protein sequences. because one gene segment, one part of your DNA, can actually be translated and transcribed into hundreds of different proteins, which have very different functions in different parts of the body. And then there's, of course, a lot of resources for cataloging things like phenotype or disease. So like the HPO is the human phenotype ontology. There's the disease ontology, which I haven't put on this slide. And all these terminologies are there to help annotate the data that is coming out of sequencers, out of microarrays, and out of gene expression, RNA-seq data. All of these things kind of need to be annotated so that we can merge them together and understand them. So these resources contain a ton of information. So within the resources themselves, there are catalogs of hundreds of millions of protein sequences, lots of molecules, diseases, phenotypes. But more importantly is what they have been used for. These resources have been used to annotate a significant portion of the data that has been generated in the bioinformatics domain. So there might be millions of entities, but there's billions or more of these annotations. So for example, there's more than 7 billion Go annotations. Those are relationships between genes and function. And if you think of, I think humans have approximately 20,000 to 25,000 genes. 7 billion annotations means that each of those genes is being annotated to, I don't know, I guess like, how many zeros is that? Probably like tens of thousands of functions or even more. Similarly, there have been other types of interaction networks that have been created from the entities within these ontologies, things like protein-protein interaction networks, pathways, and phenotype annotations. And of course, these ontologies, these terms have found their way into all levels of our biomedical infrastructure. I put 3 centers here that are part of the National Institutes of Health, but it is the same in Europe, in Asia, and other continents. There is a real demand for ways of organizing this data. And some of you might be familiar with these tools. So something like MESH terms, the medical subject headings, or the Unified Medical Language System are used to catalog data in PubMed. And then there are ontologies as part of the OBO Foundry, which may also be used here for recognizing entities in the semantic scholar corpus. And now my research focuses on a subset of,
Sorry, just a quick question. To what extent is this fragmented by the fact that these are English and these things are called national? Because proteins and genes are universal, but the Europeans have a French ontology or whatever? Or are we all in the same international one?
Yeah. So I think this really depends on what subdomain of biomedicine you're talking about. So for clinical terms, which are terms that are being used in hospitals to annotate medical records, those terms tend to need to be translated more because a hospital needs to operate in a certain language. So we have codes like ICD-10 or SNOMED, and there are efforts in each of these countries to then take the English or international version, as we call it, and translate it to the various languages of the user countries. But then things that are more for bioinformatics, things like the gene ontology or UniProt, those tend to not be translated as much because the English is being used widely as the language of research being done in that domain. I hope that clarifies a little bit. But yeah, and then of course some of these ontologies natively will have synonyms in other languages. So that it's work that is done by the actual ontology editors and curators. Okay, so my research focuses on a set of this data, and it's on a type of data called pathway data. And pathways are models of biological process. And what they are kind of networks of interactions between genes, proteins, DNAs, molecules, nucleic acids, and other things in the body. But they're not just kind of random connections. Pathways focus on biological function. So you'll have a pathway, for example, describing a metabolic process, such as how sugar is broken down in the body. or you can have signaling pathways that talk about how a hormone transmits information into a cell and changes gene expression, or you can have pathways that discuss how disease occurs. So you can think of these as essentially like a type of graph data where all of the entities or all of the nodes are these various biological entities, and all the edges are the relationships between them. So I guess now I can talk a little bit more about my research and how it relates to these pathway data sets. So the first thing I'll talk about is kind of an automatic prediction of instances for ontology classes, and the ontology I'm talking about is the pathway ontology. So this is the purpose of the pathway ontology is to discuss various types of biological pathways and how they're related to one another. Now, a single pathway is kind of an individual module within the system that's related to a certain function. But these pathway modules are actually all interconnected. The boundaries tend to be a little bit arbitrary, and they're defined in a way that is maybe useful to a scientist, but they're not necessarily obeyed in the body. So this ontology, traditionally when we want to map data sets to ontology terms, all of the annotations are done by hand. So for example, those 7 billion gene ontology annotations, those were all, I wouldn't say all, but the vast majority of them were generated by some human curator who looked at a gene and determined what function it performed based on maybe microarray experiments. So the pathway ontology has a number of data annotations to existing pathway databases like KEGG or small molecule pathway database. But we also wanted to generate new annotations between other pathway databases and the pathway ontology. So in some recent work that I did there, we created kind of a semi-automated pipeline for inferring these class instance annotations. So the curation pipeline just injects a predictive model in front of the traditional curatorial review process. So instead of having a curator look directly at a pathway and trying to determine what pathway class it fits into, we use the existing class instance annotations to train a model to predict the best class matches for each pathway instance. And this is just a shout out to the curators at the Rat Genome Database Project who helped perform and review the annotations coming out of this model. So the way that this was done, so there's quite a bit of, so maybe for some context, the size of these databases, there's usually thousands of pathways in these databases, but there's sometimes not a very clear one-to-one mapping between a pathway and a pathway class. So we took some of the training data out of the pathway ontology itself, the various class mappings to KEGG and small molecule pathway database, and also used some mappings from the Unified Medical Language System as our training data. And we also kind of used a simple logistic regression model to bootstrap additional data from one of the databases that we were trying to align with, trying to map to the pathway ontology. And that was Reactome.
Lucy, can you say again what a pathway class is?
Sure. So the pathway ontology is essentially a hierarchy of pathway classes. And each of those classes, it might be something like a carbohydrate metabolic pathway. That is a class that then contains instances of actual carbohydrate metabolic pathways from these various databases. So the ontology itself consists only of classes and annotations to instances. But in order for the data from these various databases to be kind of organized under the same hierarchical tree, then the pathway data instances need to be associated with a particular class.
How many classes are there?
There's approximately 2,600 classes in the pathway ontology, of which approximately 20% of them describe altered or disease pathways.
And each pathway belongs to only one of them?
Each pathway generally only belongs to one class, but it can be related to other classes through regulatory relationships. So also within the pathway ontology are other relationships, such as this pathway is a part of another pathway, this pathway regulates another pathway, and so on.
Yeah. Is the hierarchy very flat? So does each individual instance belong to a single class, or is there also a tree hierarchy on top of the?
It's a tree hierarchy, so if a pathway belongs to a certain class, then it really is also a member of its parent class, and so on. It depends on what part of the tree you're looking at, but I'd say this ontology is not particularly deep. I think the max depth is something like 12 layers. Yeah, but on average, it's more like 5 or 6. Cool. So had a bunch of training data, and then we trained some neural network models to essentially compute match scores between pathways and classes in these databases and the pathway ontology. And I'm not going to go into detail here, but I'm happy to discuss later. We were able to use the descriptive information on the pathways and pathway classes, as well as the pathway data itself, so the various entities and relationships that are in the pathway to help generate match scores. And then here's just an example of what an output set might look like. So for the reactome pathway for apoptosis, here are the ranked outputs from the model, and as you can see, the top 3 recommended pathway ontology classes are clearly apoptotic pathways. But if you look further down the list, p53 signaling is very much relevant to controlled cell death as well. And the pathways for cellular detox, humoral immunity, and cell-mediated immunity are all related to the innate immune response, which are also highly related to cell death. So we were able to select some classes with relevant content, and now the curators have been able to generate instance class mappings for several of these other pathway databases. So then I want to talk a little bit about pathway network alignment. So these pathway data need to be composable. Like I said, the boundaries of these pathways can be fairly arbitrary. But when we want to study some sort of biological function, we might want to take pathways about different things. So if a disease affects metabolism as well as the immune response, then we would want to take pathways for both of those functions and merge them together. So these pathways need to be composable not only within one pathway database, but also between pathway databases, because some databases will, say, only have metabolic pathways, and some will only have signaling pathways, and so on. So we need a more unified representational schema. But this is not very straightforward. So here's an example of a single pathway called the glycolysis pathway from 4 different databases: HumanCyte, KEGG, Panther Pathways, and Reactome. And this pathway is quite straightforward. It's a linear pathway that converts the glucose sugar into pyruvate, which is a 3-carbon sugar, generating some energy. And it's a linear pathway that consists of approximately 10 reactions. And this is a very well-studied pathway, so I just want to say that we know exactly what happens in this pathway. But if you look at the way it's represented in these 4 databases, there are quite a number of differences. So each of these entities is it is mapped to a term in an ontology. So for example, all of the chemical species, so the entities in the green boxes and the yellow circles, those would be mapped to identifiers in ChEBI, or chemical entities of biological interest, or PubChem IDs or something like that. And then the proteins, which are next to the blue circles, would probably have corresponding identifiers in protein ontology. But if you look at the identifiers that are being chosen, even that is not necessarily the same. So in this case, just looking at pyruvate, 2 of these resources, human psych and panther, have chosen a different ChEBI ID than KEGG and reactome. So if you were to look at these entities between the 2 resources, it's not it's not as straightforward to say that they're the same molecule. And similarly, there might be missing or extra reactions or entities. And if you look down here, because these reactions are, have direction, the direction specified between the reactions can also drastically alter the topology of these pathways, making it not very straightforward to merge them together. Yes?
Are these systematic differences, or are they, would you consider them to be annotation errors?
Some of them are annotation errors, and some of them are systematic choices made by the editors. So for example, in human psych, I would say that these 3 arrows are not necessarily incorrect. They are based on information specified in a different database about the natural direction of these reactions. And of course, keep in mind, almost all biochemical reactions are reversible. So they operate in both directions depending on the environment and circumstance. So for example, the enzyme commission database probably specified the direction of these arrows based on some predetermined pH. But if that pH is slightly lower or slightly higher, the reaction would actually operate in reverse. So this is just something that, so this would be like a systematic difference between this database and all the rest. But here, I would say this is just an annotation error because they've chosen to do that for 2 of these reactions, but have not for the 3rd, even though it's fairly, even though they're trying to observe some sort of convention.
Is there sort of a 3rd category where maybe this sounds like a very well documented pathway, but in cases where the science isn't really clear yet, how are you able to distinguish that situation from the others?
I don't know if I'm able to distinguish it, but I think I put up this as an example because it's so well studied and it still has so many problems. So if we go into areas where the science is unsettled, I would venture a guess that we would probably see more problems.
It might not be obvious.
Where the problem even is. But I don't think this is unsurmountable, right? I think there's more similarity than differences between these pathways. So we can still try to align them. And I have been working on ways to align these pathways programmatically, not just the same pathways between different databases, but also pathways that may overlap between those databases. So I've been using cross-reference identifiers to ontologies, various ontology lookups, trying to explore using lexical features, so if 2 entities have very similar names or components, and then also using network topology to infer additional alignments between nodes that have very different names, which is actually quite common. So like I said, for the ontology lookups, we're essentially using a semantic distance measure that's coming out of the ontology. So for example, if 2 ChEBI identifiers are related within the ChEBI database, then they're going to be closer or more similar than if they're 3 hops away, or if they're like 10 hops away. And then also integrating this graph alignment algorithm, the graph edit distance and evolutionary algorithm, into the alignment process to try to generate additional matches. So here's just an example of what these various techniques add. So here I have a reactome pathway called phenylalanine and tyrosine catabolism, and a human psych pathway called tyrosine degradation, which is a portion, which describes a portion of the reactions that are found in the reactome pathway. So if you just look at the cross-reference identifiers specified in both of these databases, then only the green entities match, which is really not a lot. You could pick up 2 random pathways, and there would likely be a similar number of matches, because some of these are like trivial entities that are found in all pathways, things like hydrogen ions or oxygen or water. But if we use the ontology semantic distance measure, we get a lot more matches. So these entities are in fact related, or can be found to be related in these other databases. But if you see on this diagram here, there are a bunch of squares and circles that are labeled CX and RX. So CX are complexes and RX are reactions. Now these things generally do not, these nodes generally do not have any identifier that can be, that they can be annotated with. So it's much harder to figure out what they mean. And in fact, what happens here is, although I've overlaid this pathway manually on top of the other one, there are some differences in the way that the reactions and complexes are represented. So reactome definitely represents a complex, but human psych sometimes does not. So you'll see these proteins directly leading into the reaction. However, we tried our best to use kind of a graph alignment to generate some additional matches. So running a global graph alignment gives us some positive matches, which are the entities in light green, but it also generates a bunch of mistakes due to the differences in topology in these reactions down here. So this is something that I'm still trying to explore and trying to incorporate other features. Yeah.
To what extent do you care about matching individual entities versus complete pathways? Like in your previous example, it seemed like you care about these 4 pathways are the same, right? Mm-hmm. And then match the entities afterwards. Whereas this approach seems to be matching entities within the pathways themselves. Like, is one easier or more desirable? Or?
Well, in the third part of the work, I essentially want to combine these 2 things. So matching, knowing which pathways are kind of representing the same function is important, but then being actually able to collapse them together and use them together is also quite important. So this part gets at like how do we match entities within the various pathways. So yeah, I guess that leads to essentially generating better pathways for enrichment analysis.
Is there a way to have quantitative analysis on your results? Like, for example, in this line of research, do people usually do quantitative analysis?
There's generally some quantitative analysis. I would say that I don't have quantitative analysis for this because it requires a massive amount of manual review, and we don't have, I don't have enough data to share for how well we did. But that is something that we're working on. Yeah.
The previous one for the instance of ontology classes? It's easier.
Yeah, so I do have some quantitative results for that, which I can share afterwards because they're just like a hidden slide in my presentation. I'd say, so we did a review of approximately 100 pathways and all of the annotations that were found for those, and found that I think approximately 80% of the subset we looked at had relevant class recommendations from the pathway ontology. And the remaining 20%, upon curatorial review, we found that those classes were actually just not, there were no good corresponding classes in the pathway ontology, and we needed to introduce new classes and new relationships to handle some of those pathways.
And this dataset was from the same domain that, or the same ontology that was used to train the model, or was it a new ontology?
This was from the knowledge base, from the Reactome database, which was, we did bootstrap some of the kind of like easy matches from Reactome to use as training data. So you could say that we definitely touched the database, but the point of this task is to generate an alignment, and I think it's, I think because curatorial review is required at the end to actually make the match between pathway instance and pathway ontology class, that's an acceptable thing to have done. Yeah. So this is work that is in progress, but I am still trying to finish. So we know that gene mutations lead to disease. If we think of DNA as essentially a long series, like a long series of three-letter words, then it's pretty clear how point mutations or deletions and insertions can lead to drastic changes in the way that our genes are expressed. However, most diseases are actually not caused by these gene mutations. You might have perfectly normal copies of genes, but they express in a way, or they are converted to protein in a way that leads to disease. So very common complex diseases like heart disease, hypertension, diabetes, most forms of cancers fall within that category, where they're called polygenic or complex diseases because there is no single gene that is responsible for causing them. And we'd like there to be a single gene, because if there is a single gene, then we can run experiments. You take a bunch of people with the disease, you take a bunch of people without the disease, measure their gene expression levels, cluster them together, and find out which genes are responsible for causing disease. But that's, it doesn't really work like that in reality. In reality, we need to, genes kind of operate in modules. So you can have, for example, I can have the same disease as someone else, but for my gene expression, you might see differences in gene A, the level of gene A, but for the other person, you might only see differences in the level of gene B, C, and D. However, we kind of can get at where the boundaries of these modules. Pathways are sort of one type of gene module. They're all the genes associated with a particular function. So one technique that biologists have come up with dealing with this issue of most genes are not monogenic is to essentially use pathways to aggregate the effect of individual genes into gene modules. This is called pathway analysis. However, choosing different pathway databases can really drastically alter the results here. If you think of the pathway data set you choose as essentially like a basis set for, the analogy I like to use is like for signal decomposition, then you really want the pathway data to be, you want them each to be fairly independent, but you also want broad coverage of all biological functions. So one of the things I'm doing now is essentially combining the other two phases of my work, which is identifying semantically similar pathways and then merging and aligning them to generate a set of normalized pathways, which would be more ideal for this pathway analysis application. And I will be comparing those normalized pathways against standard pathways in standard pipelines for pathway analysis, like gene set enrichment analysis. And I don't have any results to show you here. This is work that's ongoing, but I'm happy to discuss any details if people are interested. So then I'd like to talk a little bit about where I'm headed next. So the natural extension for my current work, the gene ontology has recently introduced these causal activity models, or GOCAMs. And these essentially link a bunch of standard gene ontology annotations, which are very simple relationships between a gene and a function, and link them together into what are essentially these simplistic pathway models. The benefit here is we can then take advantage of the more than 7 billion gene ontology annotations to both construct these models and also verify them. So I am collaborating with the GOCAM group on converting between kind of base pathway representations and these GOCAM representations, and also integrating some of these pathway alignment algorithms into the way that GOCAMs are curated. And I'm also interested in extending some of the previous work I've done here at AI2 with Semantic Scholar. So at AI2 as an intern, I constructed an ontology matcher which incorporated natural language text into the matching process. And an ontology matcher takes in essentially two structured terminologies or ontologies and generates a mapping between them. So a natural extension of this work might be to adapt it to map data between Semantic Scholar and other literature corpuses like Microsoft Academic Search, A-Minor, or the Internet Archive's newly introduced FATCAT catalog. But this would involve essentially matching other types of entities like authors or papers or venues, things like that. Some of which have identifiers. So for example, some papers have DOIs, some authors have ORC-IDs. So it would be also kind of analogous to some of the graph alignment work, the pathway alignment work that I was talking about before. Another thing that would be a natural extension is to link relationships found in the literature to relationships found in these various ontologies. So for example, in this sentence from a paper, it's talking about the relationship between the TRAF2 protein and the CD40 protein and a binding relationship between them. So if you go to the Reactome resource, you can find this triad, which talks about the complexing between two complexes, one involving TRAF2 and one involving CD40, into a bigger complex involving both of them. So this triad kind of is a reference to, or I guess this text above can be seen as a reference to this complexing relationship. And the benefit of trying to link these relations to these relationships that have been defined in these resources is that in the resource there's actually a lot of context around this particular triad. This relationship is part of a much larger pathway that talks about cytokine signaling in the immune system. So now we would know not only how these smaller complexes came to be, but also how they're related to immune signaling. And another thing I'm interested in doing is extracting results and methods from the literature. I think over the summer with the hackathon project, Spacey Bio, I worked on a small component of that, which was a PICO text classification model. And PICO is a model for evidence-based medicine. P stands for patient, I for intervention, C for comparison, O for outcomes. And S is often tacked on to the model, and it stands for study design. And all of these aspects are important to identify in a piece of literature to essentially determine its fitness for use in the clinic. So does this literature add to the existing information we know, and should the outcome be used to determine how we treat a particular patient? So of course outcomes are very important because that's the essential nugget of a paper, but I'm actually also quite interested in extracting the study design. And I think the study design generally tells us how much trust to put into an experiment. Of course, the type of study that is talked about the most and probably provides the highest level of evidence are these interventional clinical trials, where you take a bunch of people, you randomly that are the same, and then you randomly assign half of them to a treatment and half of them to placebo and see what happens, right? But the truth is, interventional clinical trials actually make up a very small portion of the trials that are actually conducted. And there's a variety of other trials, both retrospective and prospective, that provide differing levels of evidence. And it's important to know like where on the evidence hierarchy these stand.
Question. So this is, I understand the retroactive, so the older trials were mostly not randomized, but I'm surprised. Are you saying that it's also true for new ones?
Yeah, so interventional clinical trials are expensive, and there's a lot that we can learn from doing other types of trials. So, for example, a bunch of these retrospective trials, you can go back and look at patient records, and you can generate hypotheses, often based on them, that can then be tested in maybe retrospective studies or interventional trials. But for example, EHR studies, something I care a lot about. Like, there's if you can identify patient groups using records that have been previously generated, and you can detect some outcomes there, and then use that to form a hypothesis that you want to test, right? Like if you identify a drug interaction that's causing some other symptom, you can then test that in a different trial. And then another thing I'm interested in is semantic querying of the literature. So how can you take a natural language question and break it down into a structured query? So I think identifying ontology terms and relationships is one way of potentially doing this, is to parse this question into a more abstract representation between these various ontology terms, and then trying to identify identify articles that match the query. And it's not quite straightforward. I would say like a question like that, the important thing is to try to identify like the intent of the question. So here it's really looking for a mechanism of function rather than just a statistic on whether or not the statement is true. Yeah.
This is an idea that we've talked about a lot at S2. Can you give your kind of motivation for supporting asking this type of question in natural language versus asking this type of question in a less ambiguous format, like the parsed version, like show me the mechanism between gene X, Y, and so forth?
Yeah. I think the benefit of asking this in natural language is because sometimes it's very hard to formulate, or it's much easier to formulate these questions in natural language. And we have, I guess, like keywords that clue us into what we want. I mean, this question can be asked in ways that you can make subtle changes to this question that drastically alter what you actually want as a result. You can say, does BRCA1 cause breast cancer in humans? Or how does? Or how much does BRCA cause breast cancer in humans? So I guess you can try to come up with more structured ways of asking those questions, but you really need to predefine like all the structured ways you can ask a question. And I think there's a lot of subtlety in what people might be looking for.
You at least have to define that in the system that executes this query once it's been parsed as well. Yeah, but I definitely see some advantage in offloading the work of translating the question I have in my mind into a query language. But yeah, there's just a lot of possibility for ambiguity and error in that process that
Right. And sometimes you might be asking a question that really has no answer. In which case, you can still, I guess, like, use this to maybe suggest like an alternate question to ask or something like that.
Yeah, sorry, a follow-up question related to that. I was really interested in the annotation relation between BRCA1 and breast cancer, and you could imagine that that could refer to, you know, potentially some of the pathways that you talked about earlier, or it could be more generalized. Right. And I guess sort of overall question is, does the ontology help us think in a structured way about like different ways of representing that relationship? And then if not, I guess is that a potential area of research? So I can imagine you might want to offer like choices in terms of the results of like how you're thinking about that relationship, so that you can, you know, further qualify your question.
Right. So I think the ontology component that might be interesting and applicable here is that many of these relationships in ontologies, they're only defined between different types of entities. So this one is not obvious, but for example, like if you're looking for a regulatory relationship, that can only occur between pairs of genes, or it can't really occur between, like, you know, a disease and a gene. It just doesn't work. So knowing the type of relationship can maybe help us filter down the type of question it is and what types of answers to respond to. But I'd love to brainstorm more about how to do that. Okay, and then also another part of this work that I'm interested in maybe pursuing are some clinical natural language processing applications. So biomedical text, of course, also includes, besides the literature, also includes a lot of clinical notes, and the ITHS provides access to UW medicine researchers these clinical notes, which are generated not for research use, they're generated for clinical use, but have a lot of value for research. As I mentioned before, those EHR studies, the electronic health record studies, are a type of retrospective study that can provide a lot of ideas for future trials. And it does turn out that the majority of these clinical records are unstructured text. So the big box is unstructured text and the small box is structured. And the structured data is largely information that's relevant to administrative or billing needs. But much of the kind of meat of the record, what the doctor is saying about you, your medical history, your lab results, and various things like that can be quite unstructured. So I'm interested in maybe developing some methods in this realm for adverse event detection or quality assurance, and then deep phenotyping, which is a way of extracting from an EHR all of the phenotypic characteristics of a patient, which then, if that patient ever gets genomic testing, provides like a really interesting set of data to like compare against. So to summarize, some of my previous work emphasized automating parts of ontology curation and annotation. I talked about some methods for pathway alignment and also ways of modifying pathway analysis to improve the identification of gene treatment targets. And I'm excited in the future to extend some of these methods to other databases and, of course, use ontologies to better understand the literature and also clinical notes. Thank you all for having me. I'd like to thank my advisor, John Gennari, the various people that I've worked with, my collaborators, and then I'm happy to take questions.